shape determining interactions in proteins. Now, hydrogen bonds along the backbone. Hydrogen bonds form between the hydrogen atoms in the NH groups and the oxygen atoms in the CO groups along protein backbone. Now, hydrogen bonds of the R groups with each other or with backbone atoms. Now, some amino acid side chains contain atoms that can form hydrogen bonds. These can connect different parts of a protein molecule. Now, often hydrogen bonding side chains are present on the surface of a folded protein, where they can form hydrogen bonds with surrounding water molecules. Now, if we look here at some of these forces, you see here we call these backbone hydrogen bonds. Here we have side chain hydrogen bonds between peptide groups. Here hydrogen bonds between peptide groups. Here we have hydrophobic interactions, water-free pocket. Notice that these are the R groups of non-polar amino acids, whereas here this is the OH of the amino acid called tyrosine, and this is the CO of methyl aspartate. Here we have again hydrophobic interaction between valine, two valines. Here we have hydrogen bond between side chains, as you can see, and also we have something called salt bridge. If you have aspartate residue with methylene lysine residue positive with negative, they form the so-called salt bridge. And here we have also hydrogen bonds side chains, backbone hydrogen bonds. Now all these are non-covalent type of forces. The only covalent type of force is called disulfide bonds. Whenever you have two cysteine residues that are in close proximity to each other, they form this disulfide bond. Ionic attractions between R groups or salt bridges, the one I showed you. Ionized acidic and basic side chains will create salt bridges. Why they call them salt? Because positive and negative. Hydrophobic interactions between R groups. Hydrocarbon side chains are attached to each other, attracted to each other by dispersion forces to create a water-free pocket, as I showed you. Although individual attractions are weak, their large number in proteins always a major role in stabilizing the folded structure. Covalent sulfur-sulfur bonds, the one I told you, the occur between cysteine amino acid residue have side chains containing thiol functional groups that can react to form sulfur-sulfur bonds. Now we have something called the secondary structure of proteins, and an example of a secondary protein structure is called alpha helix. Now this structure is stabilized by hydrogen bonds parallel to the helix axis within the backbone. Now, from the N-terminal of a protein, the CO group of each amino acid residue is hydrogen bonded to the NH group of the amino acid 4 residues away. Here you can see these are the hydrogen bonds between amino acid number 1 and then amino acid number 4 in the residue. Now, C double bond O on amino acid 1 is hydrogen bonded. This is oxygen is hydrogen bonded to NH on amino acid number 5. Okay. Then oxygen with hydrogen here. Amino acid number 1 with amino acid number 5. 1, 2, 3, 4, 5. There are 3.6 amino acid residue per turn. If we consider this is a turn, this is a turn, so we have 3.6 amino acid residue per turn, and this is the regular alpha helix. Repeat distance or the linear distance here, the distance and between here and here, between the corresponding points on successive turns, the distance from here to here or from here to here, this distance is called pitch of the helix. and is equal to 5.4 angstrom. Now on the right here we have, this is the designation for right-handed helix, and this is for left-handed helix. Again here, this is another look at the alpha helix. Again, you see these are the hydrogen bonds and they run parallel to the alpha helix axis, etc. And here we have 3.6 amino acid residue per turn here, and we have from here to here the pitch of the helix, and this is equal to 5.4 angstrom. Now the second type of secondary protein structure is called beta pleated sheet. They could be two types, parallel and anti-parallel. Now it consists of two chains, as you can see. If they run in opposite direction, it is anti-parallel. If they run in the same direction, it's parallel. Now the R group here, as you can see on the amino acids, alternate one below and above. This is above the plane and this is below. This is above, this is below. This is above, this is below. So the R group alternate up, down, up, below and above the plane on the sheet. Now the hydrogen bonds here, which I shows you, stabilize interaction between neighboring protein chains. And the hydrogen bonds, as different from the alpha helix, run perpendicular or on the direction of the chains. In alpha helix, we said hydrogen bond, they run parallel to the alpha helix axis. Here no, they are perpendicular to the direction of the chain. And this is the zigzag structure that results from forming hydrogen bonds and the presence of R group one time at the top, alternating up and below the sheet. This is called beta plated sheet, you see. This is called beta plated sheet. Here you see the R group, the hydrogen bond, the amino terminal, carboxy terminal, amino terminal, carboxy terminal, etc. There are two types of proteins. Fibrous protein is a tough insoluble protein whose protein chains form fibers or sheets like this. Example, مثلا wool, hair and fingernails are made of fibrous proteins known as keratins, which are composed of alpha helices twisted together into small fibrils that are in turn twisted into larger and larger bundles. Now, natural silk and spider webs are made of fibrion composed of stacks of beta sheet. The R groups must be relatively small, so fibrion contains regions of alternating glycine and alanine. Don't worry about this. Don't worry about the fibrion here, just worry about this paragraph. Now the other type of protein called the globular protein. By the way, the fibrous protein is water insoluble, and it only gives structural role, whereas globular protein is water soluble protein whose chain folded in a compact spherical shape with hydrophilic groups on the outside, and it performs their structure which vary widely with their functions are not regular. Sections of alpha helix and beta sheet are usually present. The hydrophilic side chains are on the outer surfaces of globular proteins, account for their water solubility, allowing them to travel through body fluids. And many globular proteins are enzymes. You remember at the beginning, in part 1, I showed you a table with the some types of proteins and their functions. Some of them are enzymes, hormones, etc. Enzymes are also globular proteins. Now the overall shapes of globular proteins represents another level of a structure called the tertiary structure. As I will show you, this is myoglobin, a globular protein, and this gives us the third level of a protein structure called tertiary. tertiary structure, as we will see. Tertiary structure, the way in which an entire protein chain is called and folded into its specific three-dimensional shape, is the protein's tertiary protein structure. Each protein molecule folds in a distinctive manner that is determined by its primary structure and results in its maximum stability. A protein with the shape in which its function in living system is known as a native protein. Now, a protein can exist in many shapes called conformations, but not all of them are active. The active ones are called native protein or native conformation. A portion composed only of amino acid residue is a simple protein. Tertiary structure is drawn in a style that shows the combination of alpha helix and beta sheet regions, the loops connecting them, and disulfide bonds. As you can see here, in a tertiary protein structure, you find alpha helix like this. You find beta sheet, you see, whether they are, you see, this and this, they are anti-parallel. You can find connecting loops like these in purple, and also in yellow here and here, you find the so-called disulfide bonds or disulfide bridges. They make like a bridge between two chains. By the way, this is a protein or an enzyme called ribonuclease, and this tertiary structure was determined by X-ray crystallography. Now, conjugated proteins are aided in their function by an associated non-amino acid unit. The oxygen carrying portion of myoglobin has a heme group embedded within the polypeptide chain. A single polypeptide chain of 153 amino acids and include a heme group. It consists of 8 alpha helical regions. The nonpolar or hydrophobic residue are found in the interior or the core of myoglobin, and the polar residue are found on the exterior, as I showed you the structure of myoglobin before. Now, there is also something called quaternary protein structure. Is the way in which two or more protein chains aggregate to form large ordered structures. Now, if a protein consists of one subunit, it has a tertiary structure. If it consists of more than one subunit, yani two subunits and more, if it's called dimers, 3 trimers, 4 tetramers, and so on, then this will have a quaternary protein structure. Now, polypeptides are mainly held together by non-covalent forces, but covalent bonds and non-amino acid portions may also be incorporated. So the type of forces that tend to stabilize the quaternary structure are all non-covalent forces. Now we talked about myoglobin as a protein, as an example of protein with tertiary structure. Now let's talk about hemoglobin as an example of a protein with a quaternary structure. Hemoglobin is a conjugated quaternary protein composed of 4 polypeptide chains, each of 2 polypeptides called alpha chains and beta chains, held together by hydrophobic interactions, and 4 heme groups. So it is a tetramer of 2 types of subunits, alpha and beta. And the formula is alpha 2, beta 2. Now the alpha chains each consist of 141 amino acids, and the beta chains with 146 amino acid residue. Now the heme groups each contain an iron. So hemoglobin, the oxygen carrier in red blood cells. In the lungs, O2 binds to Fe+2 so that each hemoglobin can carry a maximum of 4 oxygen molecules. In tissues, however, in need of oxygen is released and CO2 is picked up and carried back to the lungs. This is the structure of heme. This is the structure of heme and hemoglobin, a protein with a quaternary structure. Here we have the polypeptides are shown in purple, green, blue, and yellow, with their heme units in red. So the one in red here in the middle, these are the heme groups. The heme group is like this unit. This unit is called a heme unit, is present here of the 4 polypeptides in hemoglobin. The heme consists of a protoporphyrin 9 plus iron 2. So this is our heme. لاحظوا it has 6 coordination. 4 of them coordinate with the 4 nitrogens of the protoporphyrin, and the 5th here attaches to nitrogen of histidine, and then the 6th position is left for binding oxygen. Now again, collagen now is the major constituent of connective tissue. Collagen is fibrous protein, but it is an example of quaternary protein structure, because the basic structural unit of collagen is called tropocollagen. عبارة عن 3 intertwined chains of about 11,000 residue, blue, purple, and yellow. Imagine each one consists of 1,000 amino acid residue. 3 of these coiled chains wrap around one another in a clockwise direction to form a stiff triple helix in which the chains are held together by hydrogen bonds. Then in collagen, it's stabilized by hydrogen bonds. All collagens have a glycine every 3 residue, and prolines are hydroxylated in a reaction that requires vitamin C. So every 3rd amino acid residue in collagen is a glycine. So glycine constitutes up to 33% of the amino acids in collagen. And also you can find hydroxyproline, which is formed from proline by a reaction catalyzed by enzyme called hydroxylase, which requires vitamin C. That's why deficiency of vitamin C will lead to formation of fragile collagen. And again, this is the structure of collagen, you see. Chemical properties of proteins. In protein hydrolysis, peptide bonds are hydrolyzed to yield amino acids. Digestion of proteins in the diet involves hydrolyzing peptide bonds in the stomach and small intestine, where the process is catalyzed by enzymes. Amino acids are absorbed through the wall of the intestine. A chemist might hydrolyze a protein by heating it with hydrochloric acid. So, inside the stomach and small intestine, proteins by digestive enzymes H+ are hydrolyzed into the corresponding amino acids, and these amino acids are absorbed through the wall of the intestine. However, in the lab, inside our body, we use enzymes. In the lab, we use HCl, and we heat a solution of the protein with HCl, and then we hydrolyze it to its constituents of amino acids. Denaturation is the loss of secondary, tertiary, and quaternary protein structure that leaves primary structure intact. Solubility is often decreased by denaturation. Enzymes lose their catalytic activity, and other proteins are not able to carry out biological function when denatured. This is a native protein. This is a denatured protein by heat. Most denaturation is irreversible, but renaturation is accomplished by recovery of biological activity. All the information needed to determine protein shape is present in the primary structure. Misfolding of proteins leads to abnormal secondary and tertiary structure that comprise the function of the protein. Heat. The weak side chains attraction in globular proteins are easily disrupted by heating. These are some of the means by which proteins can be denatured. Heat. Mechanical agitation. Detergents. Even very low concentration of detergents can disrupt the association of hydrophobic side chains. Organic compounds, polar solvents interfere with hydrogen bonding by competing for bonding sites. pH change. Excess H+ or OH- ions react with basic or acidic side chains in amino acid residue and disrupt salt bridges. And finally, inorganic salts, where high concentration of ions can disturb salt bridges. And this will be the end of part 2. Thank you.